Hello students, welcome to the biology class. Today we are continuing with our chapter Biotechnology, its principle and processes. In our previous session we studied about the basic concepts of biotechnology, the tools for the biotechnology, and today we are going to discuss our first tool, that is your restriction enzymes. So restriction enzymes are called molecular scissors because they used to cut the DNA at the very specific point. So which are responsible for cutting the DNA? They are present in bacteria to provide a type of defense mechanism called as restriction modification system. So they are present in the bacteria in a different form of the strains, and they are giving the defense mechanism that is called as a restriction modification system. The first restriction endonuclease, that is HIND2, was isolated by Smith, Wilcox, Cayley from Haemophilus influenzae, that is the name of the bacteria. It was used to cut the DNA molecule at the particular point by recognizing a specific sequence of 6 base pair, and that particular site which are having a 6 base pairs, that is A, T, G, C, C and so on, known as recognition sequence. So what is the recognition sequence? It is a particular point which is having a specific sequence of a 6 base pairs. So the restriction enzymes, they are commonly called as the molecular scissors. They are responsible for cutting the DNA at a very particular point, and that particular point is known as your restriction enzyme. So before we did the naming of restriction enzyme, first we should learn about the mechanism of action of endonucleases. So before the action of endonucleases, we should know the types of restriction enzymes. So restriction enzymes belong to the class of enzyme called as nucleases, and they are of two types, exo and endo. Exo means they cut the DNA at the end, whereas endo, they cut at the specific position within the DNA. So within the DNA, when they are used to cut, they give some sticky ends, and when they cut at the end, they comes out as a blunt end. And the recognition sequence of endonuclease, they are palindromic, means that is the sequence of the base pairs read the same on both the DNA strands when orientation of reading is kept same. For example, if you read from forward to backward or from backward to the forward, the reading orientation that remains the same, that pattern is known as the palindromic pattern. For example, 5' to 3' we are having G, A, A, T, T, C. Similarly, when you read from backward to the forward, that is from 5' to 3' again, the same pattern is there, that is G, A, A, T, T, C. So these 6 base pair sequence which are having the palindromic sequence, that is known as the recognition sites. So the naming of the recognition sites and the restriction enzymes. So how you will name the restriction enzyme? The first letter of the restriction enzyme is derived from genus name, and the second 2 letter is from the species name of the prokaryotic cell from where the enzyme are extracted. The Roman number following the name show the order in which the enzymes were isolated from bacterial strain. The example is EcoR1, very commonly used restriction enzyme, is derived from, that is the first letter E comes from the genus, that is Escherichia coli, CO that comes from the second 2 letters from the species RY and 1,3. So R is the strain and the number is 13 that comes out as the Roman number 1. So ECOR1, that is the naming of the first restriction enzyme which is derived from Escherichia coli R1 RY13. The similarly Hind2 from Haemophilus influenzae RD, BAM-H1 from Bacillus ameloleacaceae H, ECO R2, again that was the strain was there. So by this we can name the various restriction enzyme depending upon their genus, species. Then you can say we are having the Roman number following the name, show the order in which the enzymes are isolated from the bacterial strain, and by this we are known with 900 restriction enzyme which we are using for carrying out our recombinant DNA technology or various experiments of the biotechnology. Now, mechanism or the action of endonuclease, or you can say the restriction enzyme. So every endonuclease inspect the entire DNA sequence for palindromic recognition sequence. So first it has to inspect and find out the recognition sequence of a 6 base pair which is palindromic. On finding the palindrome, the endonuclease bind to the DNA. It cut the opposite strands of DNA in the sugar phosphate backbone a little away from the center of the palindrome site, but between the same base on both the strands. So this results in the formation of single-stranded overhanging stretches at the end of each strand called as the sticky ends. The sticky ends facilitate the action of enzyme DNA ligase by readily forming hydrogen bonds with complementary strands. So in genetic engineering, DNA from the different sources are cut with the same restriction enzyme. So very important is, so the DNA from different sources cut with the same restriction enzyme. So the same restriction enzyme is used for cutting the different sources of the DNA, that both DNA fragments have a same kind of the sticky ends. So these sticky ends are complementary to each other and thus can be joined by the another enzyme or the tool of recombinant, that is your DNA ligase, end to end. So here you can see the diagram or the steps how the mechanism of endonuclease will work out. The enzyme cut both the DNA strand at the same site. So this is the same site with the help of a same restriction enzyme, that is your ECOR1. They cut the DNA between the base pair, that is G and A. So here you can see and you will find the sticky ends. So this is GC and here is again that is the same recognition sites are there. So the sticky ends are there and the fragment, they are joined at the sticky ends with the help of the enzyme that is known as your ligase. The enzyme which form the recombinant DNA by joining the DNA fragments at the sticky ends is known as your enzyme ligase. So that name of the enzyme is ligase, so which helps in making the recombinant DNA. So this is the mechanism. So steps in the formation of recombinant, the action of restriction enzyme, the known is your ECOR1. So this is very important. Please go through once again that what are the steps. First, we are having a two, you can say the DNAs. One is from the vector site, fine. Another is the foreign DNA. With the help of the same restriction enzyme, they have to inspect or search out the recognition site which is palindromic to each other. Then with the help of the ECOR1, they cut at the same site by having a sticky ends, then joined with the help of a enzyme ligase to form the recombinant DNA. So this is the action or mechanism of your restriction enzyme. So the next is the very important topic is separation and isolation of DNA fragment by the technique that is called as the gel electrophoresis. So gel electrophoresis is a technique for separating DNA fragments so based on their size. So firstly, the sample DNA is cut into the fragments by the restriction endonuclease. So you will get the various fragments. And in this diagram or the figure, you can say this is the agarose gel electrophoresis. So here we can have a trough-like structure in which we place the agarose as the medium, which is in the form of a gel. We are having the pits, or you can say the wells are there, in which we will put the uh, sample of the DNA, in which we will have a sample of the DNA, and then we will attach the cathode and anode. The current will start, they start moving towards the point that is towards the positive side, because the DNA is negatively charged, and they will move towards the positive charge. The larger, which are of a heavy molecular weight, they are at the, you can say, towards the well side, and the smaller they will move farther, and then we can extract from them. So see the step one by one. So what we are having, so the DNA fragments are being negatively charged, can be separated by forcing them to move towards the anode under the electrical field through the medium or the matrix. And commonly used matrix is agarose, which is natural linear polymer of D-galactose 3,6-N-hydro-L-galactose, which is extracted from the seaweed. The DNA fragments, they are separate out, resolved according to their size because of the sieving property of the agarose gel. Because of the sieving property of the agarose gel, since smaller the fragment size, farther it will move. Now when they have separated out, you cannot see that fragments with the naked eyes because they are being blended with the agarose gel. So you want to visualize them. For that, we put the stain on the gel, or you can say in the medium or the matrix. So that stain is called as, or the dye is called as ethium bromide. So separated DNA fragments are visualized after staining the DNA with ethium bromide, followed by exposure to the ultraviolet radiation. The fragments are seen as the colored bands, that is orange colored bands. So separated bands of the DNA are cut out and extracted from the gel piece. So we have to take out that particular orange band or cut out from the gel. Like that we are taking out the something from with the help of a spoon. So this step is known as elution. The purified DNA fragments, now they can be used to form the recombinant DNA, which can be joined with the cloning vectors, and they can multiply in the things. Fine. So this is about your gel electrophoresis. So today we will discuss two topics, or the two, you can say, the mechanism. One is the action of your restriction enzyme, and the second is the separation and the isolation of our DNA fragments by the technique known as the gel electrophoresis. They both are very important. Please go through them and consult your NCERT for the same. Till then, stay home. Thank you and have